All right. Next up, we have Malina Loeher, who completed her PhD in marine science in Dr. Wargo's lab at the Virginia Institute of Marine Science in 2024. She currently works jointly with the USGS Western Fisheries Center and the Alaska Pacific University's Fisheries Aquatic Science and Technology Lab. Malina, go ahead. The floor is yours.
Great. Thank you so much. I'm super excited to join everyone today. So let's see, got to just figure out how to advance my slides. There we go. So Andrew gave a great overview for IHNV and the system, and we're going to dive a little bit more specifically into IHNV's host jump. So IHNV is co-evolved with sockeye salmon, one of the Pacific salmon species, and considered to be endemic. IHNV is considered to be endemic in the same range, which here is roughly indicated by this blue ellipse. And the viral genogroup of IHNV that's associated with sockeye as its endemic host is termed the U genogroup. So as commercial aquaculture expanded in the Pacific Northwest, particularly rainbow trout aquaculture, IHNV emerged in its new host, rainbow trout. And trout shares a lot of the same endemic range as sockeye, but unlike salmon, has a pretty intense land-based aquaculture industry associated with the species. So this purple ellipse here indicates the emergence of a new clade of IHNV really associated with trout farms, and this clade of viruses is termed the M genogroup of IHNV. And as Andrew shared previously, this M group, particularly in IHNV, is really problematic for the industry and can result in over 90% of mortality, particularly among juvenile fish. Oops, there we go. So we've been really interested in this genetic diversity and what we can kind of expect from IHNV in the future and how it's evolving. Here we have the phylogenetic tree of IHNV and how IHNV-U and M genogroups are organized. Down here on the bottom, I have the U genogroup bracketed by U. So this is the ancestral IHNV group associated with sockeye salmon. And U gave rise to the M genogroup on top here, bracketed in purple. And we've been really interested in how the viral phenotype has changed through this host jump from sockeye to rainbow trout. So in other words, when a virus jumps hosts, how is the phenotype going to change and what can we expect from IHNV in the future? So that's the whole theme of this talk today, and we'll give you I'll give you a little bit of a background of some of the past virulence and shedding work that we've done, and then the larger project that I'm really excited about. So our previous work investigated virulence before and after this host jump, and we conducted a set of matching in vivo assays to examine how viral shedding kinetics have evolved alongside virulence. These studies were 30 days long, and we used rainbow trout for all of them. Using our rainbow trout host, we looked at the number of fish that died, and for each one of these M and U isolates, we looked at survival. We also looked at the number of individuals shedding in each isolate treatment over 30 days and how much they shed, or the intensity of that shedding over time. So this is just a little snapshot of what those data looked like for every isolate. We did these for each of 15 isolates in IHNV. And what we found were that the emergent isolates seem to have increased virulence. So we documented the continued increased virulence among the M isolates in this novel host. So here, the blue, I've tried to keep coloring mostly consistent. Each one of these isolate bars on the bottom is a different virulence metric for an isolate. And then we have cumulative percent mortality on the y-axis. All of the blue bars are the U genogroups, and all of the purple or pink bars, depending on your screen, are those M genogroups, which seem to be increasing through time. They're organized by order of their collection year here. This is in line with farm observations, though this is kind of cool to see in this selection of just 15 isolates or 10 M isolates. And our next question was if the M shedding phenotypes are evolving in a way that could explain this virulence change as well. So we focused on intensity and duration of shedding as possible mechanisms, and we saw a very mild increase in the intensity of shedding, looking at the peak period of shedding through time, and then also duration, which here was a proportion of the positive days sampled. So over that 30-day period, the more days that we detected positive virus in the water, the higher that proportion was, and we used that as our metric of duration of shedding. So this was kind of cool to see, and this led us to our main research question for today's presentation: is how are M's continuing to evolve through time? So among those first 10 isolates, we observed increased virulence and increased shedding duration, maybe some increased intensity over time. And these isolates were selected to represent evolutionary time as well as the dominant isolates on the farm landscape. But that was a lot of variation to just try to pack into 10 individual isolates. And IH&V, we know, has distinct lineages that have arisen within the M genogroup. And these are called subgroups, IH&V subgroups. And so we wanted to know if these lineages are all exhibiting the same evolutionary trends, or if there's more nuance to be understood among these different subgroups that might present different challenges to host fish. So I'm going to bring you back to that phylogenetic tree. This is just that top part of the tree with that purple bracket, the M genogroup. And I've added a little bit more labeling detail here. Hopefully you can see my cursor, but on the bottom we have the M genogroup, MN group, and it's not a true subgroup, I think, but MN immediately arose after the host jump into rainbow trout, and then subsequently evolved into the subgroups MB, MC, and MD, which you can see on the upper branches of the tree. So for this next study, we selected an additional 30 isolates among these different branches within the M genogroup to fill in the temporal gaps of our original array of isolates from about the 1972 to 2019 was our range. And we carried out virulence and shedding assays in the same methods as we had used previously to be able to compare isolates to each other. So just a quick little snapshot of what these studies look like. You'll see a lot of tank photos among all of our presentations, but you can ignore the metadata details of all of our isolates. This is what our virulence challenges look like. We have group immersion challenges and then the shedding assays that match them to answer our questions about survival and shedding phenotypes. And from our data that we gathered from these, we modeled the virulence for our 40 M isolates, 30 new ones and our 10 previous ones. And we observed a general increase in virulence through time for all of the subgroups, which was consistent with our previous results. It's subtle, but over the course of decades, that could result in significantly more mortality if you're looking at a dense population. So our natural next question was, what is driving this change, and could increased shedding be part of that equation? Could increased shedding be related to this increase in virulence? And we first looked at shedding intensity. So among our 40 isolates, this was just looking at our peak intensity in that acute period and looking modeled across time from the 1970s to the 2010s. MN is separate here because it only represents isolates from 1970 to about 1980, the early 1980s, and then our subsequent subgroups are representative from the time period of the late 80s to almost present day. And we definitely didn't see an increase in shedding intensity among these groups, and maybe there's a little bit of a decrease among these instead. So shedding intensity didn't seem to be the main explanatory variable for increasing virulence. And so looking back at our raw data, okay, so I'll walk you through these plots. I'm going to throw up a couple different plots here, and then we'll look at them together. Here, each point is an isolate. Oops, yeah, each point is an isolate, and I put our virulence metric that we used previously on that plot on our x-axis here. So the x-axis is consistent across each of these panels, where the further to the right a dot is, the more virulent it was. And on the y-axis, we have different shedding metrics. So the first one is the number of fish that we're shedding, or the frequency of fish that are shedding. and then followed by peak intensity, which you can see that like slight decrease over time, and then what I call post-peak intensity, or a later time point of shedding across the kinetic curve. And the panel that I think is most interesting among these is this first one, where you see a general positive trend of virulence being positively correlated with increased shedding frequency. And so high virulence seems to at least correlate with shedding frequency across these decades and subgroups of M isolates. So a few takeaways here. Virulence does seem to be increasing across all the M subgroups. They have slightly different virulence measures, but seem to have the same general rate of increase from our data. And this isn't necessarily due to shedding intensity, but rather shedding frequency and duration seem to be key to figuring out like the mechanics of viral fitness. Something that I think is really interesting in this system is like we do see a dose response in some of these experiments, and I'm excited to hear from Hannah and Isabel later. I put up a couple little teasers for their work to kind of point you in that direction, but there's a lot more to be learned about IH&V in the future. These are my last slide, just acknowledgements for my lab team, collaborators, all of our funding sources, and I'm happy to take any questions if there are any.
All right. Thanks, Malina. Excellent talk. Really cool stuff. Dave Groman would like to know which strains of rainbow trout were used. Were they Idaho aquaculture strains, and any comparison with less aquaculture selected populations?
We used rainbow trout from Idaho, and the exact strain I'd have to kick over to Andrew to fill in the details, but we chose the strain, or we identified a strain that we thought would be that we didn't think was selected on for any particular metrics, and we thought would be representative of rainbow trout approximately at the time of host jump. And I'll pass it over to Andrew if there's any other details that I'm missing that you think we should add.
No, that's right. I mean, you were from Trout Lodge, and so these were unselected. We intentionally chose like a line of fish that would have been unselected, so they weren't a line that had undergone any aquaculture selection, to try to control for that. You know, what you're talking about is like, yeah, maybe there's a difference if it's an aquaculture, to get as close as we could to like the host jump. I mean, you can't go back in time, right? We wanted to get a line of fish that was as closely representative of the fish at the time of the host jump that we could. So yeah. And the same with the sockeye. I mean, the sockeye were also just less options there, but they were being like an unselected line as well. Yeah.
Excellent. Any more questions for Malina? All right. Thanks, Malina. Enjoyed your talk.